the spinal cord, we know where, again, it's anatomy stuff. We know where it ends. I mentioned this earlier, like gray matter is where you're going to find your cell bodies, white matter is where you're going to find most of your axons. Okay, so now the pathways here. So the two principal ascending pathways, the posterior column, medial lemniscus. What sense is that involved in?
Fine touch.
Specifically, what kind of touch?
Fine touch.
Yeah, fine touch. Yeah, exactly. And spinal thalamic tract is involved with pain and non-discriminatory touch.
The posterior column was fine touch and proprioception.
Mm-hmm.
Yeah.
Spinal thalamic does pain and
pain
temperature, so everything pain and temperature.
Mm-hmm.
Which are similar sometimes.
And so with these two, I mean, it would be best, I think, to go through her slides and go look at these specifically. You need to know where they decussate, that the posterior column medial lemniscus is going to be decussating in the medulla at the medial lemniscus, and the spinal thalamic is going to be decussating at the level of the spinal cord. And the spinal thalamic one can also rise a couple spinal levels too. That's why if you get a lesion in one side, you can still maintain functionality in this.
What's it traveling up and down in segments?
There's a tract.
There's a tract, yeah.
Which is already mentioned in this slide somewhere.
Yeah,
Yeah,
the Lassaris tract, yeah.
So Lassaris tract will let you go up and down.
Yeah.
Sorry, bad question.
And so as far as lesion goes, Brown-Séquard syndrome, that was the hemisection of your spinal cord. So you definitely need to know what will happen in that case, and what will happen will be based on where they decussate. And actually, I skipped the corticospinal tract. That's the one that's for your motor. We already know this again from anatomy that your upper motor neuron is what's going to decussate. So that's going to decussate at your pyramid of your medulla. So your medulla, you're going to have your corticospinal tract and your medial lemniscus tract are both decussating there. So when you have Brown-Séquard syndrome and you have a hemisection of your spinal cord, those two are ipsilateral, right? Mm-hmm. As far as what it's going to affect, yeah. So it's going to affect it'll affect motor, and it's going to affect fine touch on the ipsilateral side of the body from that cut. And yeah, so just go through this, know what the lesions will affect.
If you draw them out a couple of times and you cut the lines off, you can see what tracks you cut. So on a test, if you're confused, just draw the corticospinal tract going from the cortex, decussating in the medulla, then going straight down. And if you cut that on this side, then obviously anything coming from the left side of your brain is not working for you guys to switch around. So the left side of the brain is no longer functioning on the right side. Right? If you do the same thing with spinal thalamic tract, you can draw it. You can see where it decussates at the spinal level, go up. So you cut on this side, everything will be fine because you already crossed over. Right? Yeah.
And stringomyelia, that's just where you have an inflammation of your central canal. And so anything that's going to cross by there in the anterior white commissure, which is spinal thalamic. So anything that's going to cross there is going to be severed. But again, with the spinal thalamic tract, because it can raise in Lassaris, we can go up a few in Lassaris tract. It won't necessarily be completely wiped out. So they would have to specify in a question how far it cut and things like that.
Just an easy question. Which layer is the lateral horn? You guys remember that? Mm-hmm. Perfect. Remember quiz? Yeah. So you have to know that, right?
Yeah.
So 1 to 6 is which horn?
It was 6.
Dorsal. Perfect. There you go. Okay. And what's surrounding the central canal?
10.
Perfect. So know those. Okay. That's like an easy 1 mark.
Yeah. Right? And I'll just really quickly go through reflexes now. We're gonna get back to these when we go to shift. So a stretch reflex, that's just your patellar reflex. That's monosynaptic, and that's really important to know because I remember there was a question on one of the quizzes about reflexes, and your flexor reflex, that's like your pain. That's if you touch something hot and you pull away from it, and that is polysynaptic. So there's going to be an interneuron involved in this. But with a stretch receptor, that's just monosynaptic, just goes to your spinal cord right back out to your muscle. So it's important to know how many neurons are involved in each of those two. And yeah, that's pretty much it.
Perfect.
Cool.
All right, so I'll be going over cranial nerves. There's 12 of them, in case you didn't know that.
What?
Yeah, I know. So we'll go through them pretty quickly for the simple ones. So overall, cranial nerves are divided by modalities. There's general sensation, special sensation, afferent, efferent, and then there's visceral afferent, visceral efferent. So just know those, okay? You can go through some. We'll go through each one. We'll talk about special functionalities for each ones. So olfactory nerve. Now that's your special sense because you're smelling. We know smell is an important sense. We'll come back to olfaction later on anyway with Dr. Spielman. So we'll go with that again. But olfactory nerve, how it works is basically you smell something. It goes up to your nose. And then the first thing it contacts that has the receptors for smells would be your olfactory epithelium or the olfactory mucosa. That's where the cells are located. Now, when they say olfactory epithelium, they say epithelium, but technically they mean the skin there. They don't mean the neurons itself are epithelial-based. They're not. They're neuronal-based. So it's neurons in your nose that are being contacted by the smell or the stimulus, which are then causing a reaction to happen. So that signals back to your mitral cells. So it goes to that's your olfactory tract. Basically, that's what's going to be making that. So your olfactory nerve is going to go back into your frontal lobe, right into your telencephalon. It doesn't go through the thalamus, and that's basically olfaction. Now, the thing can happen is it can go back to your amygdala, so it can project to your amygdala, and that's what creates that emotional response from smell. So grandma's cookies would fall under that. Okay, so that's olfaction. Yes.
Your olfactory nerve is just that, like, it's really small. Like, what's going back to your brain, that's your olfactory tract. Your olfactory nerve is that initial nerve that's going through your cribriform plate. That's
it's very small. Yeah.
It's your axons of your receptor neurons.
Okay. Good. So the second one, optic nerve. Now this is special sense again because you're again seeing, and seeing is believing. So how it works is your optic nerve. So think of it just simply. Okay. So if you have your right visual field, so whatever you see in your right eye is going to end up on your left side. First of all, start with a simplicity like that. Everything on the right side of your eye is going to end up on the left side. Right field of view. I should say right field of view. Whatever you see in the right field of view is going to end up on the left side of the brain. How that happens is a little more complicated. So what happens is your right field of view, which is going to include your right from this side. So whatever you're seeing on the nasal side of the right eye. So your nasal of the right eye is going to see this right side, right? And your temporal side of the left eye is going to see the right visual field. So both of those have to end up here. So that must mean that this must cross over somewhere. And that crossing over happens at the optic chiasm. So if you can kind of picture it, if you draw a right visual field and you draw a left visual field and you get them back to the opposite areas, then technically you have to cross one of them over to get there. And the only way to do that is the optic chiasm. So up to the optic chiasm, that's the optic nerve, it's called. And then back from the chiasm to the lateral genicular nucleus is the optic tract. And then from the lateral genicular nucleus back to your primary visual cortex is your optic projection.
In her slides, she has the whole thing
drawn
out with like purple and green or whatever. Yeah. Those are really good to just practice drawing it out and practice knowing where lesions are. And things like that.
Perfect. So just only the nasal one crossover, exactly. So if you want to measure just nasal crossing over, then that's an easy way to do it. If you just draw them out a couple of times, it'll make sense. Just know right visual field ends up in the left half, and that's why when you see the block and the key thing, that works out that way. Anything else? So you got your reflexes here too. We'll talk about like what happens when you see things. Obviously, we'll have light shining in your eye. That's going to be transduced back into your brain and then respond accordingly. Because when you shine someone's light in someone's eye, they constrict their pupils, and that is a reflex that involves the signal coming in from the optic nerve and then a response from the other nerves. We'll talk about that when we get to that, which is next one. When you go back to the V1, there's a retinotopic map, just like there's a somatotopic map. So each signal for the different like rods and cones projects to a different area, kind of like telling you what you see. Okay. Yeah, what wavelength you see. Sorry, I should say. Oculomotor. Is that good for optic nerve? It's not that complicated. Oculomotor, on the other hand, is a little more. It's got the general visceral efferent as well as the general sensory efferent. We know it's only motor, and they're both efferents, which makes sense. The somatic efferent is the simple one where basically you're controlling the movement of your eye. So your extraocular muscles, as well as the superior palpebral fissure, whatever it's called. Levator palpebral superioris. So it's controlling some of the extraocular muscles, as well as the levator palpebral superioris. Now the only one that it's not controlling is the lateral and superior oblique. So superior oblique is the one that's going to make you turn medially, and because the lateral rectus makes you turn laterally. That one's easier to remember. So it doesn't do those two, but besides that, it does all the other extraocular muscles, as well as the levator palpebral superioris. Okay? So that's the general somatic efferent. And that basically signal goes from the oculomotor nucleus, goes to your eye, and tells your eye, go up, or like look up, and that'd be your superior rectus contracting. Okay? Then general visceral efferent. So how that works is basically it's going to control your pupil sphincter. So your sphincter pupillae, it's going to be able to constrict it and open it wider. It's going to have your ciliary muscles being controlled through that, and it's going to be able to like make your accommodation happen. And those are coming from your Edinger-Westphal nucleus. So your Edinger-Westphal nucleus is the parasympathetics, that's actually using the oculomotor nerve to hitchhike its fibers on. Okay, so far so good. So what will happen is you'll get a stimulus from the optic nerve saying it's really bright, constrict your eye. So what will happen is your optic nerve will project back, and then it'll go to a prepariform or pretectal. Pretectal area. It'll go back to your pretectal area. So picture this. Picture two tracks coming off the optic nerve. One going to your lateral geniculate nucleus, which is going to be interpreted into like a visual stimulus. The other one going to your pretectal area. It crosses bilaterally, goes to the edge-Westphal nucleus, where it responds accordingly how much light it saw. So when it's too much light, the Westphal will fire and say constrict your pupils, and the fibers will hitchhike back on the oculomotor nerve. So they come on the optic nerve and they go back on the oculomotor nerve, and then it tells your eye constrict your pupils if there's too much light. On the other hand, it will tell you if something is coming closer to you, accommodate your lens and relax the ciliary muscles or constrict those ciliary muscles. Constrict those ciliary muscles. Constrict. It's always constriction.
And it's important to know like which areas they travel through, like how you mentioned that it travels through the pretectal area. That was a question.
And remember, it's bilateral, so when you shine light on this eye, you're getting constriction in both eyes. So even though you're not shining light on this eye, you're getting constriction in both eyes. And that's some of the questions they'll throw at you. So the bilateral crossing happens in the pretectal area. So good so far? I mean, those are the kind of questions they kind of want you to apply. If you understand the concepts, then technically you can just draw the lines out. It's not going to be very hard for you. Okay, well, I don't need the slides, I guess. Pupillary light reflex, we already did that one, just so you can look them over. They're already written out for you. Visual reflex, this is the second one. So what do you do when something gets close to you? Not only do you actually constrict your pupils to make sure you have acuity, you also accommodate your lens as well as put your eyes closer together. To do that last one, you actually have to understand the signal. So that's going to come back from your primary visual cortex. So your primary visual cortex is going to see what's coming at you. It's going to be like something is coming closer. I want you to move your eyes closer. And remember, that's going to project back to your oculomotor, and then it's going to go forward and be like, do those extraocular muscles to rotate medially. It's going to pull the medial rectus in. Okay, so your three responses that happens. Something comes closer to you, you're going to constrict your pupils. That's going to be through your Westphal nucleus. You're going to accommodate your lens, again, Westphal nucleus. And last, you're going to move your eyes closer, medial rectus. It's going to be through your oculomotor nerve. These are all traveling on the same oculomotor nerve, just different parasympathetics versus somatic. Okay, good. Trochlear. So now we're getting into the eye things. There's more eye things. Trochlear is the one that does what? It rotates the eye which way? Medially or laterally? Trochlear. If you try to look laterally and downwards, what's going to happen with one of your eyes is not going to be able to do that. So one of your eyes is going to straight while the other one's going to go look laterally and downwards. So you're going to get this thing called diplopia because you're going to see two different things. Okay? Abducence controls the lateral rectus. So it's going to, when it contracts, it's going to move the eye laterally. So, okay. Oculomotor does medial rectus. Jesus, that's not good. Sorry. Up late last night. No, that's not. Lateral rectus. When it contracts, it'll move your eye laterally. So this is one when you try to look right or look left. Okay. And how this works is basically it has to coordinate with the other eye. Usually when you try to look right, your other eye has to look right too. That means if you're rotating one eye laterally, your other eye has to rotate medially. So that means that it must have a communication back with the what nerve to do that? The medial rectus nerve, which is the oculomotor. So you have to communicate abducens and oculomotor together. They have to be in sync for you to look laterally, because otherwise your one eye is going to look straight and your other eye is going to look laterally, which is going to be causing the medial strabismus. We talk about lateral gaze and all that other stuff.
There's a lot of slides.
Yeah, so we'll go with that. So you attempt. We'll try to go through it simply. So you try to look to the right and see what happens. Okay. So if you try to look straight ahead, you're fine, which is good. What does that tell you? That your lateral rectus is working fine. That your medial rectus is working fine, right? You try to look to the right. What happens? Your medial rectus doesn't go anywhere. Sorry, your left eye doesn't go anywhere. Your right eye works. So it tells you the right lateral rectus is working fine, correct? Because your right signal is working fine. But this eye was supposed to rotate medially, right? So that means tell you that the communication is not working right. Because the right eye did what you wanted to. Lateral rectus did its job. So abducens fired. It told the right eye, go right. But the communication to the left eye was not working right because it didn't rotate medially. So that must mean the communication between them is broken. The communication is called the medial longitudinal fascicle. Right? Yes, a medial longitudinal fascicle. Which side is it? Is it left side or right side? It's actually just described by like what side the oculomotor nerve is on. So because it's the left oculomotor nerve not working, it'll be the left MLF. Does that make sense? You try to look to the right. Your right lateral rectus is working fine. Your abducens is working fine. Your abducens nucleus is working fine. Just the communication to the left eye is not working fine, so therefore the left MLF is not working. Okay, good.
With this one too, just looking at the middle picture there.
Start with the middle.
Because you know that you're not getting medial strabismus or lateral strabismus. You know that both all the muscles are working okay. It's just like the connection between the two that must be wrong.
Your muscles have to be working and looking straight ahead, because if one of them is not working, you'll get a slight shift because they're contracted. Both of them are contracted equally when you look straight ahead. It's not like one of them is relaxed. They're both actually contracted. So if the other one's not working, the contraction will pull it to the other side. Does that make sense? So looking straight ahead will tell you right away like which muscles kind of not working. So if you looking straight ahead, you see your left eye. That medial rectus is clearly working, but the lateral rectus is not working here. So something is wrong with the lateral rectus here. Okay? So kind of like just start to look straight ahead first, and then kind of see what muscle's not working. If you know the lateral rectus here is not working, then you got some sort of like idea of what you're going for. Either the abducens nucleus for the left side is not working, the abducens nerve is not working. So we'll see what happens. Okay? And then honestly, all you have to do is basically just try to go and look at the right one and then look at the left one. You can decipher which one is working fine, what communication is working fine. So if you look at this one, honestly, like you have to take some time with these. I mean, you can tell right away the lateral rectus is not working, but the answer for this one will take some time. Both left nucleus and left MLF. And how can you tell that? Because let's just look at the answer, and now we can just figure out what the question was asking. Okay. So if the left nucleus is not working, if my left abducens nucleus is not working, what will happen? When I try to look straight, my left eye is going to go to the middle because the lateral is not contracted. That's exactly what we see, right? Does that make sense? Yeah. If the left MLF is not working, that means that communication doesn't happen when the right abducens nucleus contracts, right? Or the right abducens nucleus fires. The right abducens nucleus is going to be like, I'm going to look to the right. So your left eye should also look medially at the same time. So if your left MLF is not working when you try to look to the right, the left one shouldn't do anything, right? And first of all, you know that the nucleus is not working, so it's not going to work. So
just practice.
Just practice. Honestly, and then we go through more of them. I mean, they're all here for you if you just think about it for a couple minutes, because it's one of those things that's hard to like kind of get everyone to agree on at the same time. But if you look straight ahead, you can kind of figure out which one's not working, and then go through each step and kind of eliminate the options. Like put all the options down and eliminate which one you know for sure is not working in that step. The idea is getting the concept of how do you do these on a test, okay? So just if you practice that, then you can kind of do it.
Drawing out the pathway, like she has a slide that has like the pathway that shows the connection between abducens and oculomotor. If you draw that out, that will really help you too in understanding what's going where and that kind of stuff. So
if you draw it out the way that I did it, and then I saw which eye was damaged, if it was that eye, I crossed out that.
Yeah, exactly.
And if it was the muscle, I crossed out that nucleus.
Yeah, do the casway. Yeah, again, so just start with straight ahead, and then just go with one eye, go to the other eye, eliminate your options, and then you'll be able to figure it out. It's honestly not guesswork. It's just eliminating your options. That's all it is. And just know the left MLF is for the left oculomotor nucleus. Right MLF is for the right oculomotor nucleus. And they both communicate to the contralateral abducens nucleus.
You should also know that the abducens nucleus has an interneuron that's connected to the MLF. So if you damage the neuron, or the nuclei, that MLF's not being innervated, so that the opposite eye won't be getting the signal to, hey, you should turn your eye that way.
Yeah. So either abducens nucleus can be knocked out. That will stop the abducens nucleus and the left MLF. Or sorry, if you're talking about the right abducens nucleus, it will stop the left MLF and the right abducens nucleus. But if just the left MLF is cut, then you'll see the nucleus still working fine, but the left MLF is not going to work. So your left eye is not going to rotate medially when it's supposed to. Okay. Cranial nerves, trigeminal.
I don't know if you already explained it. Can you explain medial and lateral strabismus?
Okay, medial strabismus is basically one of your eyes is looking medially, while the other, yeah, so like what happens is, let's say you try to look right? Like, is it looking right?
No, straight ahead.
Straight ahead, okay.
Straight
so when you're looking straight ahead, you'll see a field of view, right? One of your eyes, what it's doing is it's looking slightly medial of that. So what happens is your visual field overlaps. Does that make sense? So you're kind of overlapping the medial aspects. So the medial part of the visual field is overlapping. That's what's called medial strabismus. So one of your eyes, the medial rectus is contracting, but the lateral one's not working at all. So what's happening is the contraction of the medial rectus is taking over and rotating that eye inwards. And what happens is you'll get that overlapping of the visual fields, and therefore you get medial strabismus.
Strabismus just means that one of your muscles is contracting more than the other one. So if you have medial strabismus, then your medial rectus is contracting more than your lateral.
Is it just referred to
The whole thing is
just-
called medial strabismus.
yeah, it's just-
Like diplopia is basically you're seeing two visual fields. Medial strabismus, your visual field looks like it's overlapping the medial part of it. That's just the reference of visual field.
Yeah.
So trigeminal. This one's the one that we care about the most as dentists. So you have three nuclei for this. You have your primary nucleus. You have your spinal trigeminal nucleus, as well as your mesencephalic nucleus. We already know from anatomy, mesencephalic and proprioception, so we'll talk about that in a second. So let's talk about how the trigeminal nerve is divided. Trigeminal nerve has three nerve branches: your ophthalmic, your maxillary, and your mandibular. And we know the three nerves itself have those solder lines. Do you guys remember the solder lines? Basically, they cover different parts of your face, like the onion skin effect. Okay. So what happens is, let's talk about what do we want to do first. Let's do principal sensory. That's the easy one. So principal sensory is going to be your fine touch. So if you touch finely on your face, that's basically going to go back to your principal sensory, and that's pretty easy way to do it. So any of your nerves coming that are going to do fine touch is going to go back to your principal sensory. Okay? From the principal sensory, they can take two pathways. They can take the dorsal trigeminal tract, or they can cross over. So one of them is uncrossed, which is the same side, or they can take the crossover pathway. So the one that's going to cross over is most of your reception that's fine touch is going to cross over after it goes to the primary nucleus. The only one that's not going to cross over is going to be the back of your mouth. Right? That's the only, like the inside of your mouth.
The inside of your mouth.
The inside of your mouth is not going to cross over. It's going to go to your primary sensory nucleus, travel up the dorsal trigeminal tract, and go to the VPM, which is the ventral posterior medial part of the thalamus on the same side. Okay? So that's primary sensory, and that's only fine touch. We're good for trigeminal for that one. The next one we'll do is mesencephalic, which is pretty easy. Mesencephalic, proprioception. So if you think for your proprioception, think of the muscles that you use for mastication, your masseter, your temporalis, your lateral pterygoid, your medial pterygoid. These muscles have muscle spindle fibers, which we'll talk about later anyway, and they communicate back how stretched they are by using these proprioceptive pathways. So how the proprioception is different from the other pathways is that the cell bodies, for most things that we know, are located in the trigeminal ganglion. But for proprioception, they're located right in the mesencephalic nucleus. So mesencephalic nucleus is in the central nervous system, and usually the bodies are found in the peripheral nervous system. So this is the only exception. This is the only place you'll find where the cell bodies are actually located in the mesencephalic nucleus, which is in the central nervous system. Then from there, because remember this is still one cell, that cell can go back and synapse at the reflexes. So basically it can go back to your trigeminal motor and control your mouth. So let's say you move you hit your jaw down here. Your proprioception is going to go straight back to your mesencephalic nucleus. That's where the cell body is. You haven't synapsed yet. It's going to come down and synapse at your trigeminal motor and tell your mouth to close. That's your jaw jerk reflex. So you only have one synapse. So it's monosynaptic, and basically you go back, your cell body's in your central nervous system, you synapse at the trigeminal motor and control your mouth. Okay? Clear so far? So we've done two nuclei. The last one is not complicated. It's pretty simple. So what we have is, if you can draw a box, we can divide it into three columns, and we can divide it into three rows. Okay? So how that works is basically your V1, which is your ophthalmic, which is going to be coming from like your head area and back here. It's going to synapse always in this column. V2 always in this column, and V3 in this column. If it's easier for you to remember, V1 is the most ventral, V2 is going more back, and V3 is going more back. Okay? So V1, anything from ophthalmic is always going to synapse in that column. Now, how high on that column is dependent on which part of the face it's coming from. Okay? Now, what determines what part of the face it's coming from? We divide into three parts. So if I was to look at my face, I can just divide this, and the nose part and mouth part as being the oral region. So oral, your interpolar, and your caudal region. Okay? So caudal is going to go to your caudal part of the spinal trigeminal nucleus. Your interpolar is going to go to your interpolar part of your trigeminal nucleus, and your oral part is going to go to the highest part or the most rostral part of the spinal trigeminal nucleus. And that's why, if it's V1, it's going to synapse in this column. Where it's going to synapse is going to depend on where it's coming from. It's coming from the oral area, the solder line of the oral area. It's going to synapse in this quadrant, or this, I guess, whatever, hexant. If it's coming from the interpolar, it's going to synapse in the second one. Is that clear? That's going to be your pain and temperature. So if you have pain, if you have temperature, it's going to come from there. And that's going to be where your corneal blink reflex is coming. Right? Yeah. So your corneal blink reflex is basically dependent on pain. So you touch someone's eye, it's painful, you're going to be in their eye, it's going to travel back to your V1, because you're ophthalmic, right? It's going to travel to your caudal or interpolar region, I think it's one of those, and then it's going to go from there and respond accordingly. Okay? Clear so far? That's basically it. Facial nerve, Not it's the fun one, I guess. There's a general somatic afferent, which is basically there's very few areas that do afferent for facial, mostly your ear area here. So you're gonna have your just a few things here, your outer ear, your that's about it for facial for general somatic afferent. Is there anything else for facial? That's about it. Their cell bodies are located, we already know, in geniculate nucleus or geniculate ganglia. Geniculate ganglia, which is in that ear canal, ear cavity in the auditory canal, and basically it's going to go back, not the auditory canal, I should say, what's it? In the facial canal. So it's located in the facial canal, geniculate nucleus. It's going to go back, and it's going to synapse at the spinal trigeminal nucleus. Yeah. So how that works is facial doesn't have its own nucleus specifically. What it does is it shares trigeminal spinal nucleus. Because it's such a big nucleus, it can have other things. So remember V1, V2, and V3? If you go further back, you actually have 7, 9, and 11. 7, 9, 10, and 11. So 7, 9, 10. 10. 7, 9, 10 coming back to the spinal trigeminal nucleus. So 7 is going to send its bodies to, it says bodies being located in geniculate ganglia. It's going to go back and synapse the trigeminal nucleus. Okay. SVA, which is a special visceral afferent, is going to go back to, okay, so that would be what? What would be your special visceral afferent? We know facial does taste, right? So it's special. Oh, sorry, special sensory. Special visceral afferent would be your. Oh. Special visceral afferent, special sensory afferent. Right?
No, there's also saying special sensory.
Makes sense.
Special
Special visceral afferent. So your taste. So that's your anterior 2/3 of your tongue. They're gonna go back through what nerve? Chorda tympani, and go up to your geniculate ganglia, and that's where the cell bodies are located. And instead of going to the spinal trigeminal nucleus, because it's special visceral, it's going to go to your solitary nucleus solitarius or solitary tract.
All taste goes to nucleus solitarius for every nerve. That's important.
All special visceral, basically, all special visceral afferent goes back to your solitary tract. From like 7, 9, 10, yeah, basically. So just easy way to remember that. All special visceral afferent from 7, 9, 10 will go to your solitary tract. And that special visceral afferent is mostly taste. General visceral afferent is going to control your basically your like salivatory glands or salivary glands. I don't know what to call it. Salivary glands. So you know your general visceral afferent is going to start with the superior salivatory nucleus. That's salivatory. Yeah, salivatory nucleus in the pons. It's going to go out and go to your glands. Which glands does the facial nerve innervate? Do you guys remember? Submandibular gland and sublingual gland and the lacrimal gland. Perfect. So all that is going to be done by what nerve is it? It's a greater petrosal, not petrosal. It's a greater petrosal nerve. Perfect. So greater petrosal nerve is going to go to your submandibular, sublingual, as well as your lacrimal gland, and that's going to be your tear ducts. Your mouth's going to get salivated because of the facial nerve. So if you inject someone in the parotid gland, they can't do their facial nerve. You're going to get a dry mouth, and you can also get dry eye on that side. SVE is going to be the facial motor nucleus. It's called SVE, not GVE, because it's branchiomotor. Because you're controlling muscles that are going to be branchial based. Well, they're derived from the branchial arch. In the case of facial, we know it's the third branchial arch. And it's going to be doing facial expression. So all of the muscles of facial expressions are going to be done by facial nerve. Second branchial arch, sorry. And all the muscles of the facial are going to be facial expression will be done by facial nerve. And that's going to be all special visceral efferent because of the branchiomotor. That's the only reason it's branchiomotor and special visceral efferent.
Both upper motor neuron, but when you're getting injured, let's say, your upper face, because it's bilateral, it's not going to be affected. But your lower face, it goes contralateral. It needs to cross. So then that side, one side is going to be affected. Does that make sense?
Always affected. Yeah. So if you were to cut your upper motor neuron, that would control like your upper part of your face contralaterally to both sides. If you were to cut that, you wouldn't. Your right one, you wouldn't notice a difference in the upper part of your face.
Because the left one will still cover for it.
Yeah, your left one can cover for it. You would notice the difference in the contralateral lower part of your face because that only gets it from one upper motor neuron. That's what he's saying. It's redundant.
Lowers that. If you screw over, there's a lot more. Vestibular. Okay, so 8, you have 2 parts to it. You have the vestibular and the cochlear part. Vestibular is your balance. Cochlear is your hearing. Vestibular part is actually the semicircular canals, the saccule, the utricle. Semicircular canal is going to tell you the rotation that you have. So if you rotate right, if you rotate left, we'll go into more details in the vestibular section. But the rotation is done by the hair cells. This is going to transmit back to your vestibular ganglion, which is then going to go back to your vestibular nucleus. in the pons medulla. So basically, what will that tell you is that either your head's rotating and you'll have reflexes with that. If your head is like going up and down, if you're accelerating up and down. And the great part about this vestibular nucleus is that not only does it project back into the brain, it can actually have fibers going other places. So fibers can go down your spinal cord to make your muscles oriented so you can balance yourself. It's one of those evolutionary things that our body has basically like evolved to do. It's when we see ourselves rotating our head or rotating our bodies, we can accommodate that for quicker than just by processing by the brain. Because if we process it by the brain, it might be a delay, and a delay is more than enough to knock us off our feet. So if we can process that information right in the spinal cord and just send it down to the legs, then it's much faster, and we can accommodate for that turning or that push that we just got really quickly. Okay? So it goes back to the cerebellum too, because remember, cerebellum coordinates movement, so it makes sense for it to go back to the cerebellum. The location for the vestibular nuclei, there's 4 of them: superior, lateral, medial, inferior. You don't have to know what they do, just know that there's 4. Secondary, we already talked about it. It goes to multiple ears to the CNS. And the one we're talking about later on, especially, is the vestibular reflex. That's the one that tells your eye, it's the slow reflex, it's the one that tells your eye to jump back to kind of focus on like what was happening here. So if you try to rotate to the right, what will your eyes try to do is like trying to fix their gaze on what you're looking at already, which makes sense because you're rotating quickly, you still want to catch what was happening there. So your eye will rotate with your body first, and then it will jump back to kind of gaze at the thing, and that's the vestibular reflex that we'll talk about later. Is that good? That's the vestibular. The cochlear part is the hearing. So there's 2 things the cochlear does. First thing is actually listening to sound. Second thing it does, it helps you localize sound. So it tells you where the sound is coming from. So how does it do the hearing thing? There's 2 parts of the nuclei. There's the anterior part, there's the posterior part. And what would happen is that you get your sound wave coming in. It will hit the tympanic membrane, which will then create a wave in your fluid, and that will bend your hair cells. We'll talk about it later anyway. So the hair cells will synapse to they'll have spiral ganglia. Each hair cell has multiple hair cells connected to spinal ganglia within the cochlea, and all of those will accumulate back to become the cochlear nerve. That cochlear nerve that can go into 2 parts, anterior and posterior. Anterior part is going to do most of your sound. So like actual localization, exactly. So it's going to do more of your localization. So that means that if you're going to localize sounds for the anterior, there has to be more synapses along the way for one route versus the other for it to have a delay. You want to introduce a delay in the sound so you can tell how far that sound came from. So the anterior nucleus, what will happen is you'll have a synapse here. You'll have a synapse of the superior olivary nucleus. You'll have a synapse here, which will then go to this side because it's already crossed over. That means that when the signal comes through here and synapses here, 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 it will be later than that initial just one synapse here and going up here. So that means that your brain can process what the delay was and kind of localize where that sound came from. That's the anterior. Your posterior is much simpler. Your posterior sound just comes in, synapses the nucleus, goes up to your inferior collicular, and then goes back into your medial geniculate. Because remember, lateral was for optic nerve. So your medial geniculate nucleus is for your hearing. So your medial geniculate nucleus will get the signal for the hearing, the sound that was actually heard, and it goes back to your temporal lobe where the sound is going to be processed, and that's called your area 41. But anyway, the point is basically the sound is processed there. So posterior, I actually hear the sound. What did the person say? Is it the alphabet, A, B, C, D, E? Anterior, I'm using that information that person said to kind of localize where that sound came from. Is that car coming from that way or that way? Good. More synapses along the anterior. Just know where the synapses are along the way. And posterior, less synapses.
And like the trapezoid body, just
yeah.
like the other one, just
anterior cochlear nuclei comes in, nucleus of the trapezoid body, synapse first there, superior olivary nucleus there, inferior collicular nucleus here. This pathway is called the lateral lemniscus tract, in case you want to know that. Inferior collicular nucleus here, then synapse of the medial geniculate body. It goes back here. Honestly, the slide tells you everything.
It's important to know in case they ask what areas the nerve travels through.
Exactly. Good. Glossopharyngeal. So glossopharyngeal, this vagus have the exact same modalities. So just if you memorize one, basically it's a flipping of a few words and you get the same as the other one. So GSA, general somatic afferent, your glossopharyngeal does written of their pharynx, posterior of the mouth. It does your gag reflex, so sensation for the gag reflex can go through your glossopharyngeals. GSA, every time you see S for the glossovagus and all those ones, just know that it's a superior ganglion. S and S go together, so the cell bodies are located in the superior ganglion for GSA, S, somatic, all of them are superior ganglion. They will also project, remember, spinal trigeminal nucleus, because trigeminal shares its nucleus. It's so big, so facial, glosso, Your number 10, vagus. We'll all go back to this, okay? That's it for GSA. That's as simple as it gets. GVA, on the other hand, that's doing your visceral afferent. So you want to control and regulate your homeostasis. For regulating your homeostasis, you're going to need to get the information from your viscera. And to get the information from the viscera, a specific part is your pressures. Your baroreceptors, your chemoreceptors in the carotid body and the sinus are going to go back through your glossopharyngeal to tell your brain how you're doing. If you get up too quickly, that signal's going to come through here and tell you that your pressure got lowered, pump harder. Okay?
What were you saying with the S and S thing?
Oh, GSA. So all the somatics, the genosomatic afferent, the somatic ovals are going to go to your superior ganglia. All the visceral are going to go to your inferior ganglia. And that's true for Glosso, that's true for vagus. Yeah, both of them have the superior and inferior ganglion. That's where the cell bodies are located. And for both of them, the somatics always go to your superior, the visceral always go to your inferior. Okay. So we already talked about this. This goes back to your solitary nucleus because it's visceral. All the visceral goes back to your solitary nucleus. GVE, that's going to be your branchiomotor. We know Glosso does what? Only a couple of them, yeah. So Glosso does the inferior salivatory nucleus. That's going to be doing your parasympathetics for your salivary glands, and that's going to be specifically going to your otic and going back into your parotid.
That's
yeah.
just more anatomy stuff.
Exactly, that's just more anatomy stuff. You don't need to know that. Just know that inferior salivatory nucleus is where the parasympathetics start. That's where the initiation is going to happen. You're going to hitchhike on the glossopharyngeal and travel with it, and then the rest of it basically is anatomy again. Special visceral afferent is going to be your taste. Just like facial did special visceral afferent, it's going to go back to the same nucleus solitarius. Now it does just a posterior 1/3 of the tongue. And remember, because it's visceral, inferior ganglion. Okay? Visceral, inferior ganglion. Anything somatic will be superior ganglion. Lastly, SVE, only one muscle, solipharyngeus. And nucleus ambiguous. It's ambiguous. We don't know which one does what. Okay? And glosso, vagus, accessory all use nucleus ambiguous. Making connections. Vagus, very similar. So if you look at the last slide, very similar. It's going to be very similar. So for GSA, cell body, superior ganglion, just different areas. It does ear stuff. It does a little bit of tympanic membrane. Projects a spinal trigeminal nucleus, just like the glossopharyngeal. So look at the similarities. The similarities are important because glosso and vagus are very similar. If you just know one of them, you'll know the other one. GVA, that's going to be your visceral afferent. So again, your baroreceptors, your chemoreceptors. How does your stomach feel today? How does your heart feel today? That's going to be all done by vagus. So it does a little bit more encompassing than in glossopharyngeal. And so you can read this basically. Mucosa, baroreceptors in the aortic arch, she mentions. Chemoreceptors in the para-aortic body. So it's like receptors, but in different places in the carotid body, carotid sinus. I don't know why she chose a different place in anatomy, we thought. Thoracic and abdominal viscera is going to tell your brain how they feel today. Cell bodies, visceral, inferior ganglion. Because it's visceral, nucleus solitarius. Okay, similarities. GVE. Visceral efferent starts in the dorsal motor nucleus, and we know it's going to do a lot of the body stuff, so intramural ganglion. So your parasympathetics will start in your dorsal motor nucleus, travel on the vagus, and go to the ganglion located at the organs, and those ganglions are called intramural ganglions, and that's where they will synapse. So the innervation for that basically will do your thoracic, you do your abdominal. You're controlling all the parasympathetics going to these areas because you need to be able to basically when you run or when you have when you relax and calm down and sitting back down, you need to be able to tell these things that yes, they can do their job now. Like you can work harder now. I'm in a relaxed state. Okay. SVA, your special visceral afferent is going to be your taste. It does the 1/3, like the dime shape on the back or whatever quarter shape on the back, as well as doing some of the it does the epiglottis as well. SVA, GVA, inferior ganglion, nucleus solitarius. Okay. SVE, we know it does visceral efferent because it does branchiomotor, and specifically which part it does the levator vela palatini, which is involved in your lifting of your soft palate. We know it does musculus uvulae and palatoglossus and all the pharyngeal and laryngeal muscles. So we know the pharyngeal plexus is innervated for motor by the vagus and by the somatic for the glosso. So that must mean that when you have a gag reflex, the somatic must travel through the glossopharyngeal, and it must come back. The motor for the reflex must come back through the vagus.
For those reflexes, just make sure you understand which is going to be the sensory part, which is going to be the motor part of that reflex.
Yeah, and it says which one is which. So, and then nucleus ambiguous, again, because the motor, we don't know specifically what part is done by what, but nucleus ambiguous. Yes. Accessory GSE, trapezius. It does trapezius for general somatic efferent. For special visceral efferent, that's the only kind of exception. It's weird because you know it's nucleus ambiguous, so you don't know which nerve does what. So we say sometimes that accessory has special visceral efferent because it can do some of the branchiomotor. So it can do some of the laryngeal muscles. Hypoglossal nerve, we know that's going to be moving your tongue muscle, your intrinsic and your extrinsic muscle. The only exception is your palatoglossus. And that is going to be controlled on the... Okay, so for the contralateral and the ipsilateral muscles. The hypoglossal is contralateral from the brain and ipsilateral on the normal side. Yes. So contralateral from the brain, so upper motor neuron for the hypoglossal crosses over. The lower motor nucleus is ipsilateral. So that must mean that a hypoglossal, when you think right side, to stick your left tongue out or left side genioglossus out. So if you want to contract your left genioglossus, the signal is coming from the right side. If you want to contract your right genioglossus, the signal is coming from the left cortex. Okay, so if you draw the pathways out, again, just draw crossing over for the upper motor and draw ipsilateral for the lower motor, and you can cut things off and then see what happens. That's honestly just a drawing thing. It's a schematic. And that's all the cranial nerves.
Okay, so guys, I know it's a lot of stuff for John. That's the most of it. That's the bulk of it. Okay, that's 10 questions on our midterm. So know that, right? That's actually 10% of your final quiz. That's why we spent a lot of time on the John stuff. The main points that I want to make that are going to be important is your cranial nerves. You got to know which ones into the 7 modalities that we talked about. Okay, really easy, special, visceral, and I'm going to send out a table with all of them and categorizing each cranial nerve. That's going to make it so much easier for you guys. Okay? Which cranial nerve is involved with special visceral efferent or special visceral afferent. Okay? Know that the other ones, lesions. There's going to be probably at least 4 questions on lesions, right? On this thing, on
this
that
nerve. This is
nerve.
the first speculation.
Right? Okay. But like, right? Because there are 10 questions. So you can pretty much kind of guess on what are the main concepts. So know those. If you do the lesions, then you're going to get it right. Okay, because we're running short on time, we're going to skip the last quiz 7, which is something that you guys just did. And we can talk about it at the conference anyways. So I'm going to move on to Saint Genet stuff. I'm going to give you the main pointers that you do need to know, but most of it, as you recall, it's going to be something that's factual, okay? So, okay. Just the neuron, I know you guys all know soma is talking about the cell body, dendrites, that's going to be receiving the input, axons where it's going out, and we're going to have classifications. The main point about this is you need to know what Nissl body is. What is Nissl body? Nissl body? Yeah. What is it? Yeah, sacks of rough yard, right? Okay, so that was on the quiz. Most likely something going to be emphasized on the contents of soma versus dendrites versus axons, right? So know that this one has Nissl bodies for cell bodies as well as neurofilaments, okay? Neurofibrils encompass neurofilaments as well as I think it was glial, neuroglia, I think it was, or something. So axons, these are some of the terms that you should know. This is just like one pointer. If he does put it on, you should get that one, right? Look at the last part. You should understand lemma, plasma, right? Exoplasm, cytoplasm, you know. Axon collaterals is when they're dividing. Telodendron is at the terminal, and then the boutons are at the bottom. That's the actual junction between your muscle and your axon, okay? So know that if you draw it out once, you'll get it. It's really simple. So, you know, you can have one question on that, one question on the contents of soma versus axon versus dendrites. Does that make sense? That's, you know, that's like 1 or 2 questions right there, right? Okay, so classifications. Bipolar, pseudo-unipolar, multipolar. I don't think we had a question on that one on the quiz. It was on the practice quiz that we received, but it wasn't on the actual quiz. Know that, you know, like bipolar, you'd find them in the sensory organs. You find them in olfactory when it's going from, you know, the receptor. Sorry, you find them in, olfactory as well as the rods, cones, photoreceptor to bipolar. So you'll find it there, right? Pseudo-unipolar, as you know, you find it in dorsal root ganglion.
So know the locations of where you'd find them and know what that means. Multipolar, you're going to have a lot of dendrites coming off from the cell body itself. Cool? Does that make sense? It's pretty simple stuff. Okay, so the process of how it works, you guys already know this from Schiff going over it again and again and again, how calcium is being released and how it all works. You know that when the action potential comes, there needs to be voltage-gated channels opening up so that you can get neurotransmitters being released so that it's going on the receptors of the target tissue. So as long as you understand that there's neurotransmitter release from the action potential that's coming, that's releasing these vesicles, that's encompassing the neurotransmitters, and they're going to interact with the target tissue. That is the main point of this. Okay, and it's going to cause hyperpolarization versus depolarization, which we'll talk about later, anyways, on how that membrane potential works. So that's what it is. Okay, neurotransmitters, we'll talk about those later, but you know that as for the last quiz, you knew muscarinic receptors and things that are affected by acetylcholine. If you guys remember all those, it's the same thing over again. Okay, so you guys are going to know those. Norepinephrine, which receptor is that affecting? Right, those ones that were complicated, right? Adrenergic receptors, right? Norepinephrine.
All the alpha 1, beta 1, they're all adrenergic.
Those are all adrenergic, okay?
Versus muscarinic.
Yes. Okay, so norepinephrine is the neurotransmitter. The receptor is the adrenergic receptor, right? And those receptors have subcategories of alpha and beta. Remember those? Yeah? Okay. GABA, it's inhibitory, serotonin, inhibitory. The ones that you need to know is that these are inhibitory, these are stimulatory. It's not always, but for this course, we're going to categorize them that way. Cool? Okay. So we're going to talk about PNS and CNS and how they differ. PNS, really easy. No two S cells, okay? Schwann and we should have satellite cells. Perfect. Okay. If this shows up, Schmidt-Lantermann-Claps is where you're going to have the separation because of the cytoplasm of your Schwann cells. That's an easy one.
Only seen in Schwann cells too, not oligodendrocytes.
Yeah.
Only PNS.
Right. What is the counterpart of Schwann cells in CNS? Perfect. Oligodendrocytes. All right. Okay. I know we talked about satellite cell with, I think it was muscle, but we're not being tested on muscle. So if it shows up on the exam, the satellite cell they're referring to has to be about St. Genet satellite cells. Okay. These guys are going to be covering the ganglion. They're going to be around there, and they're supporting them. Okay. So no satellite cells and Schwann cells as part of PNS. You got to be able to differentiate which cells you see in PNS versus CNS. It's going to be an easy general question, right? Okay. Where is saltatory conduction actually happening? Because that was a quiz question. The nodes of Ranvier, right? It's not actually going through in between where the myelin sheath is. Internet. Cool. Okay. What do we call the distance between one node to another node? Internode. Perfect. So know that as well, right? Intraperiod line and major period line. Perfect. So this is where your membranes of the Schwann cells are contacting each other. But major period line is when they're actually fused. So we see this as a darker, and that's why we say major period versus intraperiod. And you'll see them throughout. Okay. And mesaxon, I don't know if that was on the quiz. Can't remember. I don't think so. But what it's referring to is an open spot in the cytoplasm of a Schwann cell that are opposing each other, that are facing each other. Okay. So they're going to be open spots for the axons to be traveling in. Does that make sense? Okay. Open spot on a Schwann cell cytoplasm. Cool. Okay. CNS, there are 4 ones that you need to know. Astrocytes, see, these are the ones that are bolded. They're the most important. They're blood-brain barrier. These guys are the ones with feet on blood vessels, the ones on places where it doesn't have the myelin sheath. Remember those? And they're star-shaped. That's why they're called astrocytes, right? So the main point of astrocytes is going to be the blood-brain barrier and repair. It's probably the main ones. Okay, and removing neurotransmitters. Sure. Okay, so there's two different kinds that he talks about with two pictures that I don't know if we have them. No. Okay, so we have protoplasmic and fibrous. Okay, fibrous you'll find them in the white matter. Okay, protoplasmic you'll find them in the gray matter. Do you know which one short and branched one is? Do you guys know? What? Okay, so protoplasmic is gonna be your long branched one. Fibrous is gonna be your short ones. Okay, just know those. Did we put in the presenter notes? Yeah. Okay. Cool. Okay. Oligodendrocytes, you guys already know. It's the same thing as Schwann cells.
There's one to many.
Yeah. If you see it on I know Saint Genet didn't say that he's not going to ask about staining and whatnot, but if you see it, they're not really stained very well. The cytoplasm isn't stained; it's more clear. Versus, I think microglia, you're going to see darker, dense stained nuclei. I don't know if he's going to ask that. But microglia, the main thing, immune inflammation. So these guys are protecting. These guys are helping out. They're smaller cells, but they're helpers. Smaller helpers. Okay? Microglia. Oligodendrocytes, Schwann cells. Ependymal cells, they're going to be lining the central canal and the ventricles. Okay? I think there was a question on this. I feel like there was. Okay. Tenocyte was the projection from the ependymal cell that's going into the spinal cord. Okay? It's not going towards the central canal. So know that. The tails are going away. Okay? Okay, perfect. And they don't have basal lamina. They're kind of forming the basal lamina themselves. That's what they're doing. Okay, they're separating them. Okay, PNS and CNS structure. Okay, there was definitely a question on this. Remember epineurium, perineurium, endoneurium. I think a lot of people got it wrong anyways, so he might just put another one there. I have no idea, but this is an easy question that you should get right. Surrounding one fiber is
Smallest.
endoneurium, right? So it's going to have the myelin sheath around it, and it's going to have that endoneurium right around that sheath, okay? Right around that axon plus the myelin sheath. And then perineurium is going to be around one fascicle, okay? Fascicle. If you see one fascicle without the S, plural, then it's going to be peri. If you see fascicles as a bundle of fascicles, then it's going to be epineurium. Got that? Easy? Yeah? All right, cool. Okay. Let's see here. Okay. I think that is it for that one that you need to know. Okay. So remember these ones with Pacinian and Meissner's and Merkel's disc, and you were like pulling your hair out. You're like, why do I have to know all these? Okay, here you go, hair follicle. Know which ones have free nerve and know which ones are encapsulated. Encapsulated ones will always have connective tissue around them. Okay, and know their functions. Their location, I'm not sure if he's gonna emphasize too much on that, but for sure you gotta know if it's touch, if it's pressure, if it's vibration, if it's cold, if it's hot. Which one does the hot one out of the encapsulated? Do you guys remember?
Yeah.
Some Italian name.
Yeah, that's
Ruffini's, right?
Yeah.
Ruffini's.
I think about it because it's like it reminds me like pasta, so you have to boil to heat it up. So
Okay, I think
That's
of
not-
roofies and whatever.
Like Kraus, cold.
Yeah.
Like a German guy, cold.
Okay. Okay. So apparently German people are cold.
No.
Kraus cold. Okay. So Kraus is cold. Ruffini's is hot. Easy. Okay.
Italians are hot.
The ones with M, Merkel's as well as Meissner's, they're touch only. Okay? That's an easy one. Meissner's and Merkel's touch. Okay? Hair follicle, you're going to have pain, temperature, and touch. So the other ones, you're going to see that they have multi-functions. Okay? So hair follicle, multifunction. Merkel's, like I said, M's, just touch only. Right? So Meissner's, like I said, touch only. Pacinian, touch, pressure, and vibration. Perfect. This one's the onion-like one. If you see a lot of connective tissue around it, that's the onion-like one, Pacinian. Perfect. What did you say? Share it.
No deal with this on video.
Okay. So Ruffini's, like I said, hot, Krauss, cold. Neuromuscular spindle and Golgi tendon, you already know from the last quiz. When he said there are two types, what were the two types of spindles that we might say?
Dynamic and static.
Perfect, dynamic and static, right? Dynamic was exactly, rate of change, and static was just the length, right? Okay, Golgi tendon, you guys already know. Is it reacting to the muscle length or muscle tension? Tension. Perfect. You're teaching here. Okay, so you guys know all these, understood? Pretty easy, right? So what I want you to take away from this Scio workshop is going to be the patterns and how you're going to associate it into memory. Okay, that's why I'm telling you with M's, remember them with touch, with Ruffini, hot, and apparently Krauss, German people are close. Okay, and the other ones that are not these guys, they're going to be multimodal, like I said. Cool? Easy. Okay, spinal cord organization. Know that it's going to have stem cells that are dividing along. And he was saying that most of our organs actually do have stem cells. Before, we used to think that stem cells are only in the fetus and they're not really being produced. They're in the bone marrow. But now we realize that they're in a lot of different places, right? Okay. So you're going to have ventricular, zone, intermediate, and then marginal. Ventricular is where you're going to get the dividing multipotential cells, okay? That is the main point of this one, and you know that the mantle zone and marginal zone, they're going to become the marginal layer and the mantle layer, like we talked about before. Cool? Easy? Okay. I'm saying easy, but it might, I don't know.
Just say easy,
Yeah.
move on. That's the best idea.
Okay. So spinal cord organization, know that you can actually compare the two and so that you can actually differentiate between the two. What are you going to find mostly in the white matter? Axons, perfect. In the gray matter, you're going to find the cell bodies, right? So know those. You're going to find the cell bodies here that are more, I think it was loose, okay? In the white matter. Where does it say that? Do we have this on here? Okay, we'll add it on later. Okay. Anyways, so you know the oligodendrocytes, you're going to see a lot in the white matter because they're being myelinated. Easy. Okay. What else was there? Yeah, I think we're good here. Okay, so this one, the way I memorize it is for, medical, GP, like general practice, GP. Okay. General practitioner, sorry, or general practice, doesn't matter. Okay, so medical, GP, and medical, or I don't know if that helps with anyone. I'm just making up stuff, right? Okay, they just end up being GPs anyways. Okay, so that's that. You're gonna need to know the layers and the main ones. We have the details in the presenter notes because we didn't want to just put all of them there. What do you find in layer number 5?
Cells.
Beds. Okay, that's good. That's going to be an easy point right there that you need to know. Bed cells you find in 5, and know which ones are communicating with which layers. Okay, so for this, I would know which layer corresponds with what name. Okay, because it's going to go external and internal for granular and pyramidal. Okay, and so know which layer corresponds with the number and the name. Know which layer has which kind of cells. That is the most important one that you should definitely know. And which layer is communicating with which layer.
And Golgi type 1 and type 2.
Okay, and Golgi type 1 and type 2. They have different lengths. Golgi type 1 is longer or shorter?
Longer. Think of one being like who's faster, who's going to be number 1? So the long guy or the short guy?
Cool.
At least always number 1.
Okay, and Golgi type 2, shorter. Okay, know those, right? Okay, so cerebellar organization, it's kind of similar to this, but it's only got 3 layers, and Purkinje cell layer is just 1 cell, okay, 1 cell layer thick, okay, and we already talked about climbing and mossy fibers in cerebellum for the previous quiz. That's pretty much it. That's what you need to know. Okay, and so which ones on the periphery? Which ones at the cortex of the cerebellum? Molecular or granular? Yeah, perfect.
They're actually
Okay.
organization.
Yeah, okay. And that's the same thing with this. Okay, cortex, molecular. Okay, so molecular outside, and then Purkinje and granular inside. Cool. So get the patterns, right? For cerebral cortex, cerebellar cortex, very similar. Okay. All right. So plasticity. What does that mean? What does plasticity mean? Anyone? What? Exactly, regeneration, right? So it's actually able to regenerate. So strengthening and weakening of nerve. Okay, it's actually called neuroplasticity, where if you have trauma to one side, your brain will actually try and compensate and grow into that region. Okay, and your neurons do have the ability to do that. And when we talk about plasticity, we're talking about strengthening and weakening, but we also talk about learning and memory. It's able to regenerate and regenerate new pathways for learning and memory. Okay, cool. So how does it actually happen? Okay, so there are three things that are mainly happening. So your damage happens first, right? And then you get Wallerian degeneration. I like to think of it as I'm gonna say Wall-E. It's cleaning up the garbage on Earth, I guess. I don't know.
And we talk about it later on.
Yeah.
Like the lesions of the lower motor nucleus.
Yeah, exactly. So on the distal end, what's happening is these macrophages are gonna come and they're gonna eat it up, and they're gonna regenerate through the guidance of which cells? Schwann cells. That's one of the main points of the regeneration of PNS. Okay. So chromatolysis is when you actually get dissolution, okay, of the Nissl bodies there. So you're gonna actually notice that there's less granules inside. Okay, and the cell body kind of expands a little bit. Okay, I know it doesn't show in the picture much, but the cell body is expanding as well as the chromatolysis is happening. Chromatolysis literally means that you're losing color. You're losing color because your Nissl bodies are being dissolved, right? Okay, and your nucleus actually moves away to the periphery. Okay, okay, and Schwann cells are proliferating and they're helping out with the guidance. Good. Okay. So the main point, know those chromatolysis, Schwann cells, and Wallerian degeneration. Okay. Okay, regeneration in CNS. So one of the main questions that you might be asked is, which one has more plasticity, CNS or PNS?
CNS.
Perfect. Right? Yeah. Okay, so that's why if you get any trauma in your head, it's hard to recover from. But if you have trauma in your periphery, then you might actually be able to regenerate enough nerves to come back and whatnot. And we talk about that later as well, right? Okay, perfect. So regeneration in CNS. The main point is that in the CNS, what happens is these guys that are debris of the damage, they kind of stay there. And what they're doing is they're blocking the guidance of anything that's trying to form. And these oligodendrocytes, they undergo apoptosis, and they kind of become quiescent. So they're kind of dormant there. And so what happens is there's no cleanup so that they can form new pathways. And it's actually inhibiting it instead of, not doing anything. So it's actually worse. And microglia are going to come because they think anytime there's a damage, microglia are going to come and they're going to release inflammatory responses and whatnot. And so basically, at the end of the day, it's not good. Okay, it's not looking good for CNS. That's what you need to know. Okay, so what are the two criteria for a stem cell? This is kind of going into blood as well. What you guys say? What?
Pluripotential
Mm-hmm.
and self-regeneration.
Perfect. Okay, so it can self-renew, and it needs to be pluripotential. It needs to be pluripotential. Okay, so this neural stem cell can actually make more of neural stem cells. So it can divide itself, go under mitosis, right? And it can also differentiate into multiple things. So that is what he talked about here. What is the criteria of a stem cell? Okay, easy question there. So subventricular zone, we already kind of talked about this because we talked about the ventricular zone being multipotential and it's actually dividing. It's the same thing, except in this case, we have it in the astrocyte region right here. And we found that through research that this is the layer that's actually dividing, and that's the source of the neural stem cells. That is the main point on this. Okay? And we give you the definition here. It's the intermediate filaments for GFAP that's being released here, that's being expressed on these guys. And it has something to do with getting the neural stem cells there, but he didn't go into the details about it. Okay? Perfect. Did everyone understand that? That was pretty short. That's six questions on phage and A stuff. Okay? So I gave you some of the main ones that you're going to be seeing. I would focus on those. Those are the main concepts. Cool? Okay.
Cool. All right. So now we're going to go into a bunch of shift stuff here. All these act potentials. Okay. So equation for the Nernst potential. This is super straightforward. The thing to remember is if you want to change the Nernst potential for an ion like potassium or sodium, you need to change the concentrations. Changing the permeability isn't. You need to physically add more ions. Changing the permeability won't change the Nernst potential. The Nernst potential is an inherent characteristic of that. So there's that one question on the quiz, how would you change the Nernst potential for whatever? You need to actually increase the amount of that ion. You can't just change the permeability. But when you do change the permeability, it's going for a particular ion. It's going to the membrane potential is going to go towards that cell's Nernst potential. So if I increase the permeability for sodium, which is its Nernst potential is plus 65, it's going to go towards the cell membrane is going to go towards plus 65. That's the main big thing. Everyone knows this. ATPase. I mean, well, how many times have you learned that 3 get pumped out and 2 get brought in?
Wait, what's that?
You also need to know that a chloride ion is going to get pumped out as well to keep this electrically balanced. This is depolarization. You're having sodium channels open, and then repolarization, is going to be potassium channels. That sodium channels are going to have an extra state, open, closed, and refractory. That contributes to this refractory period down here. Yeah, as well as the ATPase. Yeah. So formula to know. I don't know how this got deleted, but this is for membrane potential. You need to know capacitance, which is the area. I think the area squared divided by thickness of the membrane. And capacitance is just something's ability to hold the charge on either side of it. So if you have a really thin membrane, then the charges on either side have a really easy time holding on to one another, right? Because there's less stuff in between those two charges. If the membrane is really thick, the two charges are really far apart, and they have a hard time interacting with one another. So you have less capacitance. Resistance, we know this. We have this time.
Applying that, there would be less capacitance at the node of Ranvier, and there'd be more capacitance. There'd be more capacitance at the node of Ranvier and less capacitance at the myelin sheath because it's so thick. That's why you don't get charges going through it.
Exactly.
That's the point of capacitance.
Yeah. And we have this time constant, which is equal to resistance times capacitance. He did mention that for an axon, capacitance is going to be it's going to be a constant.
So you assume you don't assume myelination, unmyelination, you just assume unmyelinated the whole way through. So it's just one capacitance.
Yeah. And so what's going to be changing though is resistance. And so what time constant is measuring how slow charges can move. So if you have a resistance that all of a sudden gets really, really low, like when you open a bunch of channels, your time constant is going to get really low. A low time constant means that charges are going to move quickly. So when you have right here, a lot of channels are opening, so the resistance is going to be low, so time constant's also going to be low, so it'll happen fast. When you're coming back down, though, you're having a lot of channels closing or repolarizing, and when that happens, resistance is going to go up. So your time constant is going to go up, and that just means that you're going to have it's going to be happening slowly. Charge is moving slowly. That's all that means. Nernst again, that's obvious. You know that. Definitely know these. Know that potassium's minus 80. I had 80 and yeah. Okay, so electrotonic spread. This is basically refers to the spreading of the charge within the axon. A really important formula to know and understand is this one for length constant. Length constant is describing how far a charge can travel within the axon. Okay, so it's given by membrane resistance and longitudinal resistance. Membrane resistance is the resistance, obviously, of the membrane. Longitudinal is what's inside the cytoplasm. For an axon, longitudinal resistance is pretty much going to be constant because it's just they're all going to have the same kind of contents in there. What's going to change is your membrane resistance, and that's going to change in things like when you have myelination. With myelination, the membrane resistance is going to be really high, right? Because myelin's help insulating it, right? So that's going to increase that. And so basically, what the length constant, like I said, length constant is how far this charge can spread. It's going to make it easier for adjacent like axons to fire, basically. Like I have it written here. You know that as an axon gets larger, it can transmit it faster, but you can see that as it gets larger, that the resistance is going to go down for both of them. But you know, resistance is going to go down faster longitudinally, so you'd still get an increase in length constant. Does that make sense? Okay, because that's like I said, this formula is really key to understanding electrotronic spread and how that works. So myelination, he mentioned that one way to increase speed is to just keep increasing the diameter, like I said, but with humans, we have to keep that within our spinal cord. So myelination really, really helps with that because at the same diameter as an unmyelinated axon, you can get a faster speed, and that's given by these two formulas here. And that was a question on the quiz as well. Velocity is proportional for unmyelinated. Velocity is proportional to the square root of diameter. It's also proportional to the length constant. Again, I have it written here. All right, synapses. Billy kind of already touched on this. So we know there's two ways. There's two ways that vesicles can dock: kiss and run, and fusion. I mean, I'll let you guys read over that. But the important thing is that when calcium, what is going to what's the membrane potential that's going to open calcium gated channels? -40. Yeah. So at -40, calcium will open. It's going to come in. It's going to make everything in here more neutral. So now the vesicles can actually bind to the membrane, and it'll cause an EPSP or IPSP depending on what it releases. We'll get to those. So now to return to homeostasis. Sorry. This is just that you just have to know it. You have to know. There's two ways you can restore calcium. There's a fast way and a slow way. The fast way just happens initially, and that's just sequestering in your smooth ER. And the slow method is your exchanger or your calcium ATPase. No, you get 3 Na coming in for pumping out 1 calcium. You have to get your membrane back too for the vesicles that you fused with the membrane when you let it go. I'm not going to go through this. You can just have a look at it. But you're the worst. He went over this in lecture, and it's important to understand the cleft size. Obviously, if it's narrow, it's going to be fast because it can get over that small gap really quickly. If it's big, it's going to be slow because they've got to travel very far. Ionotropic, metabotropic. He went over this again recently with the receptor types. Ionotropic is going to involving some kind of ions moving. Metabotropic is normally going to involve cyclic AMP.
So one's a G protein, so it's got another G protein coupled with it. One is just a receptor and a channel built into the same thing. That's the ionotropic. So the receptor and the channel are the same thing. Yeah.
No. Can someone tell me what does DAG do in terms of calcium? Increases. How does it increase? Yeah, it increases permeability for calcium. Whereas IP3 is releasing intracellular stores of calcium. So yeah, know that difference because that can be an easy true or false kind of thing.
Sensory transduction. Let's just go through the main concepts. Transducers are different than just regular axons. Hey guys, Jeff, you good? Good. Okay. So transducers, how they work is they don't have voltage-gated sodium channels. They'll have stimuli-gated sodium channels. So what happens is when you get more stimulus, more sodium enters the cell. More sodium enters the cell, more depolarization. So they don't have voltage-gated channels at the transducers, but further down the axon where you have voltage-gated channels, you can get an action potential there. So as you increase your stimulus, more sodium comes in until you get to the nurse potential. That's where you see the kind of that curve. Okay, that's it is. So how that works is once that sodium comes in, it will travel further down the axon because it's Brownian motion. It moves all the time. It moves further down the axon until it gets to the point where there's voltage-gated channels. And when it gets to the voltage-gated channels, then you can find an action potential because you have enough depolarizations to open enough sodium channels to get an action potential going. Okay, so So that means that if you keep a stimulus pressed for long enough, you can get a depolarization that stays long enough in the cell to cause another action potential to happen. Therefore, a stimulus can have frequency of action potential dictating its intensity, because we know action potential is all or none. So the only way to code intensity is to get frequency of action potentials. Okay. So sensory are EVSP only, so only going to be positive. Deciphering intensity is already done by action potentials, frequency, not intensity. Adaptation, so if you keep getting the same stimulus, over time the threshold's going to keep rising and rising and rising until a certain point you kind of become adept to that pain stimulus. So if you keep putting pain on someone, it's not going to be as effective as it is early on. Just in case you're doing kinky stuff. So give them rest. The Onion corpuscle. Onion skin, this is the one that really adapts quickly. It's the one they use for touch, so like vibrations and stuff. So if you put clothes on, that's the one that's going to be stimulating this. Unfortunately, you don't want to feel your clothes on your body the whole day, so it will adapt really quickly. On the other hand, hair cells of the utricle, they almost never adapt because you want to constantly know if you're accelerating up or down or sideways. Okay. Rods. So eyes. Eyes use two things, rods and cones. Rods are the ones we're going to mostly talk about. Cones are the smaller versions of rods and do color. Okay. So rods are going to be the light and dark. In the dark, you have a baseline. So that's the important thing. So they kind of work opposite of what you think. In the dark, you have a baseline because in the dark, you're actually making CGMP. The CGMP is going to open up sodium channels. So in the dark, you have CGMP constitutively being made in the cell to open up sodium channels, which makes the channels more leaky, which makes the cell more leaky to sodium. Now, if it's more leaky to sodium, it automatically depolarizes because the Nernst potential of sodium is positive 60. So that means as it depolarizes, it will get to a certain point above the threshold. It'll be liminal to keep firing action potentials. So in dark, you're firing action potentials at a certain frequency. Now, if you have light or photons hit the cell, a certain part of the cell will detect that and turn off this system, break down the CGMP, so you decrease the permeability of sodium. Decreasing the permeability of sodium will let less sodium into the cell. That means that the cell will repolarize, so therefore it will fire less action potentials when it's darker. That concept is clear. How does it do that? The special photon receptor is called. I'll just make sure I do the same slide. Yeah. So in the light. So the photon will hit this thing called retinal. It's initially in the formation of 11 cis retinal, so it looks like a triangle. When the light hits it, it becomes a trans. So it becomes straight line. It comes off the one end. Then over time, it will come off the other end. That retinal is going to interact with a protein called transducin. This transducin, what does it do? It actually goes and says, phosphodiesterase, turn on. Phosphodiesterase says, you know what, this CGMP, I'm going to cleave you. It cleaves the CGMP. That's how we get the reduction of CGMP, which we already know was turning on the sodium channels. So if you don't have CGMP anymore, you turn off the sodium channels, and therefore the permeability decreases for sodium. And that's how you get the repolarization and less action potential frequency when you have the light. Okay? Retinal was a product of retinol that was stored in the pigment layer. That's important. Pigment layer does three things. It stores the retinol, which is a precursor for retinal. Pigment layer also does the following. It absorbs the extra light that comes in. Extra light comes in, doesn't bounce around. Instead, if it misses the cell, misses the rod, it will get absorbed by the pigment layer, so your eye knows exactly where the light came from. So you get visual acuity by absorbing that extra light. In animals that are nocturnal, on the other hand, you don't have that pigment layer as much as dark, so your light will bounce around, so you get better night vision, but unfortunately you lose the acuity because you don't know where the light came from because it bounced around maybe before it hit the rod. So you lose that acuity, okay? And the third thing you will do is it'll collect all the garbage and kind of recycle that. So the garbage basically in the rod cells is those saccules. So the saccules are basically where the retinal is located, and the saccules are made in the rod cells. And as they are made, they will travel down further from the isthmus all the way down to the base of the rods, and they'll get recycled when they leave the cell. So the pigment layer, three things. It will do the retinal production, so it will have the retinal stores, it will have the pigments for absorbing extra light, and it will do the degeneration of the saccules once they're done. Rods, they work backwards. The base of them is where the actual signal happens, because the base is the rod, and then the bipolar cells for the neurons, and then back to the optic nerve. So they're kind of oriented that way. Okay, so rods are the back of the eye. Good. That's all there is. Cones, thinner, they do color, they respond to different wavelength. There's the red-orange one. So the blue wavelength is the one that the blue cone cells do. Blue. Simple as that. Blue wavelength, blue cone cells. If there's a green cone, then it will do blue-yellow light. If it's a red cone, it will do orange-yellow reddish light. Okay? So it will respond to the highest stimuli at that wavelength. So the wavelength, if it's blue wavelength, the blue cones will respond to the highest stimulus. Okay? They will have the action potential frequency increase with the right stimulus. Good? And blue and yellow together make green. That's what they're called green cones. In case. Hearing and balance. Yeah. Hearing, and then you're joking. Hearing, we already did hearing a little bit before. So this is just the hair cell part of it. Hair cells, how they're special is that they have cilia at the end, stereocilia and kinocilia. Kinocilia are really tall ones, stereocilia are the tiny grouped ones. So what will happen is when you have a sound wave come in, it will propagate that, it will move the liquid, right? And as it moves the liquid, it will push the hair cells down. So when it pushes them down, what happens is if you push them this way, you'll increase the surface area on that side. Do you agree? If you increase the surface area, what happens is more potassium channels are open. More permeability of potassium, more potassium can flow in. Now this is important because the liquid that makes up this area is called an endolymph. And the endolymph is different than normal extracellular fluid. Endolymph actually has a high concentration of potassium, which is different because normally extracellular fluid has very low potassium. The high potassium here is